Okay, let's unpack this. We are diving into the bedrock of international pharmaceutical quality today. When a new chemical entity moves from the lab bench to a registered product, the control of impurities shifts from a matter of chemistry to a matter of global regulatory consensus. This deep dive is a technical briefing on one of the most consequential documents in new drug development: how we rigorously control and critically qualify chemical degradation on an international scale. We're approaching this not as students, but as peers in the regulatory space.
That's absolutely right. Our source material is the foundational text governing quality control for finished products. It's the ICH Harmonized Tripartite Guideline Q3B R2, specifically the Step 4 version from June 2006. If you work in analytical development, regulatory affairs, or quality control, this document dictates your stability studies and final specifications. Its impact is enormous because it represents the harmonized standard recommended for adoption by the regulatory bodies of the European Union, Japan, and the United States.
And the mission of Q3B R2 is highly focused, isn't it? It provides guidance for registration applications on the content and, much more strategically, the qualification of impurities found in new drug products. And this applies specifically to products derived from chemically synthesized new drug substances that have not been previously registered anywhere.
Right, and defining the scope is the first critical step to compliance. We had to clarify what we were talking about. Q3B R2 focuses exclusively on impurities classified as degradation products. These are the chemical changes that occur during manufacturing or storage or reaction products that result from the drug substance interacting with its excipients or the immediate container closure system. If the impurity is not caused by degradation, it belongs in a different regulatory bucket.
That clarity is vital. I mean, if we are treating this as a technical briefing, knowing the boundary is everything. So what is explicitly excluded from the Q3B R2 mandate? We are excluding those process-related impurities like residual starting materials or reaction intermediates, which are typically handled under Q3A. We also exclude impurities arising directly from excipients, anything leached from the container closure system, or the entire classes of biological and biotechnological products.
And the list continues to narrow the focus. Peptides, oligonucleotides, radiopharmaceuticals, fermentation, and semi-synthetic products, herbal products, all of those are out. Finally, it's important to remember that this guideline separates out basic quality issues. Extraneous contaminants, which are GMP matters, and issues like polymorphic forms or enantiomeric impurities are also excluded from the specific mandates of Q3BR2. It's surgical in its focus on chemical degradation in the finished product.
So we know the boundaries. Let's talk about the rationale and foundational requirements. Why does a regulatory body need to see the summary? It's more than just a list of peaks, right?
Much more. The guideline requires applicants to summarize degradation products based on a sound scientific appraisal of potential degradation pathways. This isn't a retrospective exercise. It requires the development team to proactively determine how their drug substance is going to break down, including anticipated interactions with the entire formulation and the packaging.
And here's where the continuity requirement becomes a massive strategic hurdle. The impurity profiles of the batches proposed for commercial production must be directly compared with the profiles of the batches used for safety and clinical studies during development. If you introduce a new excipient or change a manufacturing step and a new degradation product pops up, you must discuss and justify that difference. The expectation is continuity of quality and exposure from phase 1 straight through to market registration.
And this brings us to the core action trigger: identification. Any degradation product observed during stability studies at recommended storage conditions must be identified when its level is greater than the identification thresholds detailed in Attachment 1. Identification is mandatory above that line.
But wait, if a new peak appears above the identification threshold and we hit a wall, we just cannot identify the structure despite our best efforts. What then? Is the application dead?
No, but the guideline is unforgiving on documentation. If identification is genuinely not feasible, the application must include a detailed summary of all unsuccessful laboratory efforts. You have to prove due diligence. Regulators want to see the scientific work, not just the failure.
Here's where it gets really interesting, though, regarding exceptions: the critical safety override. Normally, if an impurity is below the identification threshold, we don't worry about identifying it. However, the guideline carves out a critical exception. Analytical procedures must still be developed, even for low-level degradation products, if that product is suspected to be unusually potent, toxic, or producing significant pharmacological effects. Safety concerns trump numerical limits every single time. That places the burden on the sponsor to justify the lack of concern for any low-level, high-risk degradation product.
That demand for high-level technical scrutiny leads us directly into the mandated analytical rigor and reporting standards of Section 3. The application must include documented evidence that analytical procedures are validated, which means referencing ICH Q2A and Q2B. But specific to Q3B R2, the validation must demonstrate rock-solid specificity for separating and quantifying both specified known or quantified and unspecified degradation products.
To truly prove that specificity, the validation study isn't complete without proper stress testing. We're talking about samples intentionally stored under relevant stress conditions: intense light exposure, high heat, high humidity, acid and base hydrolysis, and oxidation. If your method cannot resolve a degradation product generated under stress, it's not specific enough for Q3BR2.
And the quantitation requirement is equally rigorous. The quantitation limit for the analytical procedure must be set at not more than the reporting threshold. This ensures that the lowest level the regulator cares about, the reporting level, is actually being measured with acceptable accuracy and precision. And a key analytical note: if you use the new drug substance as your reference standard for the degradation product, but the analytical response factors differ, you must account for it, either by using a calculated correction factor or by taking the conservative approach of overestimating the impurity.
Now, for the often overlooked area that causes major regulatory hiccups, the specific rules of reporting found in Section 4. Quantitative results must be presented numerically. No generic statements like "within limits." The regulator needs data, and those mandatory rounding rules, they are there to ensure absolute global consistency.
They were deceptively simple, but absolutely critical for the dossier reviewer. If your result is below 1.0%, you must report to the same number of decimal places as the applicable reporting threshold. For example, if the threshold is 0.10%, a result of 0.06% is mandatory. You can't just report 0.1%. However, once you hit 1.0% or higher, the rule changes, and you report only to one decimal place, like 1.3%. This small detail is a basic QC check for reviewers, and failing it flags the entire application for inadequate regulatory understanding.
And building on that, there is the mandate for summation. All degradation products found at a level greater than the reporting threshold must be summed and reported together as the total degradation products for that batch. The regulator is interested in both individual risks and cumulative exposure.
This clarity in reporting brings us directly to Segment 3. The heart of the guideline, Attachment 1, the specific thresholds. These are the numerical limits that serve as the decision engine, triggering action, and they are entirely dependent on the maximum daily dose, or MDD, of the drug substance.
Right, the thresholds are expressed in two ways: either as a percentage of the drug substance in the product, or as the total daily intake, the TDI, of the impurity in micrograms or milligrams. Let's start with the base level, the reporting thresholds. These are the levels above which we must even bother recording the data. If the MDD is less than or equal to 1 gram, the threshold is 0.1%. If the MDD is greater than 1 gram, the threshold is tighter, dropping to 0.05%.
The complexity ramps up significantly with the identification thresholds, where we see the key risk management principle, the whichever is lower clause. This is how the guideline protects patients taking low-dose, high-potency drugs. For very low doses, MDD less than 1 milligram, the limit is the lower of 1.0% or 5 micrograms TDI. This 5 microgram limit is incredibly protective, ensuring that for a very low-dose drug, even a seemingly small percentage impurity might trigger identification if that percentage equates to a high absolute intake.
And as the dose increases, these thresholds adapt. For the common therapeutic range, MDD greater than 10 milligrams up to 2 grams, the threshold tightens to the lower of 0.2% or 2 milligrams TDI. This shows the regulator's recognition that the risk profile changes dramatically depending on whether the patient is taking a 50 microgram dose or a 1.5 gram dose. The limit is engineered to catch a maximum tolerable daily intake of the impurity.
Finally, we get to the qualification thresholds, which are the fences above which you must definitively establish the biological safety of that specific degradation product. These are generally the highest thresholds, signaling the maximum acceptable level before mandatory safety testing kicks in. For MDD between 10 milligrams and 100 milligrams, the limit is the lower of 0.5% or 200 micrograms TDI.
And for those large doses, MDD greater than 100 milligrams up to 2 grams, the qualification limit is the lower of 0.2% or 3 milligrams TDI. Above 2 grams, it settles at 0.15%. These tiered dose-dependent limits are the operational boundaries that drive every decision about the final product formulation and packaging.
With those thresholds established, we move into segment 4, focusing on specifications and the strategic qualification process. Section 5 mandates a specific, highly organized structure for the new drug product specification, covering 4 distinct categories of degradation products.
This structure leaves no place to hide. First, you must list each specified identified degradation product with its acceptance criterion. Second, each specified unidentified degradation product that exceeds the identification threshold must also have a specific acceptance criterion. Third, there must be a general acceptance criterion for any unspecified degradation product, which must be set at not more than the identification threshold. This captures the unknowns. And fourth, the acceptance criterion for the total degradation products.
And defining qualification is the crux of the final acceptance. Qualification is the process of acquiring and evaluating data that establishes the biological safety of an individual degradation product, or the overall degradation profile, at the specified acceptance levels. The acceptance criterion you set for a degradation product cannot be higher than its qualified level.
So, how do we qualify a product without running years of toxicology? A degradation product is considered qualified if its level has already been adequately tested in existing safety or clinical studies. Or, crucially, if the degradation product happens to be a significant human metabolite already present and tested in animal and/or human studies, you can leverage that existing data. The goal is efficiency, but safety first.
But if qualification thresholds are exceeded and you lack safety data, we turn to attachment 3, the decision tree. This is not just a flowchart, it's a strategic regulatory blueprint. The very first question the decision tree poses, if a degradation product level is exceeded, is this: can the level be reduced to not more than the qualification threshold?
That is the critical insight, and it's where regulatory strategy meets chemical process development. The guideline implicitly favors process control over safety testing. If you can modify your container closure system, change your storage conditions, or refine your manufacturing to bring that impurity below the threshold, that is the most scientifically and economically preferred route. It eliminates the need for further toxicological scrutiny.
If reduction is not possible and the degradation product is unidentified, the decision tree mandates structure identification. If the structure is identified, the next step is assessing known human-relevant risk-checking databases for known genotoxicity or toxicity of that structure. Only if the risk assessment demands new safety data does the guideline suggest specific testing.
And that minimum safety screen is very specific. It usually involves conducting minimum genotoxicity studies, typically requiring a test for point mutation and one for chromosomal aberration, both in vitro, along with general toxicity studies, usually lasting between 14 to 90 days in one appropriate species, designed to allow direct comparison with previously qualified material. That is the cost of exceeding the qualification threshold.
This deep dive reinforces that Q3B R2 establishes a robust, dose-dependent, and strategically driven framework for degradation product control. For our technical audience, the key takeaways are the absolute requirement for specificity in analytical procedures, the mandate for precise numerical reporting based on those rounding rules, and the non-negotiable requirement to either qualify or reduce levels above the thresholds for registration.
It's the final provocative thought here. It's that this entire framework pushes sponsors toward proactive manufacturing control. The decision tree's preference for reducing an impurity rather than testing it means that pharmaceutical development teams should view the qualification threshold not merely as a test failure line, but as an economically and scientifically inefficient place to be. Your true goal is to invest in process chemistry and packaging to stay firmly below that identification threshold, ensuring superior patient safety and a streamlined registration application.
It's a beautifully engineered system that links trace chemical structure directly to patient safety regulation.
For you, the listener, whether you are designing stability protocols or defending your dossier in a regulatory meeting, internalizing these specific numerical requirements and their strategic implications is the foundation for successfully delivering safe new medicines to the global market. We hope this deep dive helps you navigate those complex thresholds with greater insight.